That hush is really alarming. Anyway, welcome to everybody here, members, their guests, and visitors. You're really very welcome to this lecture organized by Friends of Imperial College. Fire regulations. You can escape that way, turn right out into the main hallway, or go out of the back. When you're in the main hallway, main doors at the front, go that way out near the tower, which is where the assembly point is. In the event, unlikely event that you can't get through those doors, there's a rear emergency exit which you can use and follow. Friends of Imperial College is a independent charity that organizes lectures like this for the public and visits to laboratories, not only here at Imperial College, but elsewhere. We've got a group going this weekend to see the Large Hadron Collider at CERN in Geneva, and I'm sure there are one or two here who are going on that trip, and we wish you well, and I'm sure you'll be completely amazed by the technology and the precision that you see there. I'd like to welcome schools that are here. We've got the Jewish Community Secondary School in Barnet, which I read is set up relatively recently. It's the first cross-denominational school in the UK, and currently the headmaster is C of E, which makes it very interesting and perhaps warming. We've also got the Isaac Newton Academy from Ilford. Welcome. St. Paul's School in Barnes, and the Cardinal Vaughan Memorial School at Holland Park. They're frequent visitors and they're always very welcome. I'd like to thank the college for allowing us to use these premises and providing us with a technologist. And I also like to thank you for coming to support us. And by paying an entrance fee, you help to make sure that the administration is supported, and all the school pupils, of course, come free. It's part of what we do as a charity. I'd also like to thank Asad Ayub, who's our main sponsor this year. We, of course, support students at the college, and any excess money at the end of the year goes to the President's Scholarship Fund. So welcome. I would like to introduce Professor Melanie Welham, who is going to talk about a life in bioscience research. She's Executive Chair of the Biotechnology and Biological Sciences Research Council, the SRCs. It's one of 9 research councils that's come together under a new body called UK Research and Innovation. And the idea is, and I'm sure Melanie would talk a little about it, is that bringing together universities, businesses, research organizations, charities, and government bodies in order to create the environment for research and innovation to support and help the country. Prior to 2018, when she became chief executive, she previously served as executive director, science, and before that she was at Bath University and professor of molecular signaling. Indeed, she was the first female professor in her department, and congratulations on that. She graduated in biochemistry from Imperial College here before undertaking a PhD at UCL. So she's well traveled. I'd like to introduce and welcome Melanie Welham.
Thank you. Thank you, Rod, for that very kind introduction, and it's a pleasure to be here on such a beautiful early summer. Is it early summer or is it late spring evening? And I was struck by walking in how the blue sky and the tower and how iconic I think that sort of still is. So I'm going to talk to you about two sort of distinct parts to the presentation, really. And I've just sort of summarised here, so I just so that you can anticipate what's actually coming ahead. I'm going to start off with a very short kind of snapshot of my early career, and I do that because I think it's important to put in context what comes next, and I'll kind of refer back to it. I will then talk about some of the research that I was involved in when I was a full-time researcher at the University of Bath, and about the broader potential that stem cells actually have, and some of the aspects that we need to still need to address in research in order to realise that potential. And then I'll talk about the new challenge, I think, that I took on when I joined Bbsrc as Director of Science, as Rod said, and how that sort of change of direction has really enriched my life, and I hope to share some of that with you, to share what the opportunities are from bioscience research and why it's so important to supporting that and how it's going to help us tackle some of the big challenges actually that we face, not just as a nation but across the world. So sort of a journey, so hopefully you will bear with me. And I just sort of start off really with that sort of snapshot. Oh yes, thank you very much, Rod. So I was a fresh-faced undergraduate, and I graduated in 1985, which is a very long time ago, and in biochemistry. But my journey started before that. I was actually the first person in my family to go to university. My parents were incredibly supportive, but I think they really didn't understand at all what the university life was going to be like. I grew up in rural East Anglia, on farms. I was surrounded by the natural world. I was fascinated by the natural world, and that's why I think that I was interested in that area. But I was hugely grateful to the teachers that I had, and knowing that we have schools here, I think the teachers have a really important role to play in inspiring the pupils that they teach. And I was really guided by my biology and chemistry teachers to consider going off to university, and actually they were the ones who suggested, well, why don't you think about going to Imperial? And actually that was the best advice I could have had, because it set me up incredibly well for the rest of my career, and so incredibly grateful to them for that. I then, I think, sort of progressed through an academic career track, doing a PhD, and then going overseas to Vancouver, Canada, which, if any of you've been there, is a fantastic place. Spent 6 years there as a postdoc, developing my research interests, and then came back to an appointment at the University of Bath, my first lectureship. So that's a fairly standard academic career track. Of course, life goes on alongside one's career, and I think I actually, and this is a true story, I met my husband on my first day at university, and we're still together, however many years later that is, a very long time later. And he does remind me of that occasionally. He's not here this evening, he's overseas, unfortunately, otherwise I'm sure he would have come along. And so life goes along, and shortly after we returned to the UK from Canada, we had 2 daughters. So you're then sort of balancing a busy academic career, teaching, research, administrative activities, along with raising a small family. And my roommate from my first day at the university, who I'm still very good friends with, sent me this card for my birthday. She said, "This is how I imagine you," she said. You're kind of doing your research every day, and then, you transform suddenly. And I have to say that at times, life did feel rather like that. And I got to the point where I thought, I'm not sure how I can actually maintain kind of running on this treadmill, trying to keep all the balls in the air at the same time. And at that point, I took the decision to apply for a fellowship from the organisation that I now lead, which gave me 3 years of dedicated time that I could spend on research. So it gave the opportunity for someone else to come into the university to take on my teaching and administration. Obviously, I still had my PhD students, but I could really concentrate on the research. And what it allowed me to do was really kind of pivot the research that we were doing, we had been studying the molecular signals that regulated blood cell development. But we were just at this time in the early 2000s, the whole area of stem cell biology was really starting to take hold. The interest in it was increasing. And so this kind of really tweaked my interest, and I wanted to focus our research questions onto really, and our expertise onto better understanding stem cells, their behavior, and how we could then translate that understanding into really helping realize the potential that those cells have. So I applied for this fellowship and I was lucky enough to be successful. We no longer run this fellowship scheme, which sometimes it sits at the back of my mind thinking, we really ought to reintroduce this because it was so valuable to me. Maybe we will one day. So it allowed me to really start seriously looking at the potential of stem cells. And the model that we used and studied in the laboratory were mouse embryonic stem cells. And you see two pictures here. On your left, I think that is, you will see these are colonies of stem cells which we have stained with a particular marker which only stem cells possess. So these have probably got a couple of hundred cells in these colonies. They grow in very tightly packed colonies. And the fact that they're staining red tells us that they are stem cells. If we treat them in particular ways, and at that I've always been interested in the chemical cues and the signals that change cell behavior and instruct cells to do certain things. So if we treated them with particular chemical cues, then these cells change quite dramatically. And this is over a period of 3 days. So this is what they look like if you just grow them in the conditions that they like. But if you start to change some of those conditions, they lose this red staining. That indicates they're not stem cells any longer. They've changed to different cell types. And we were quite interested in studying this process. And I'll explain a little bit more about that in a moment. But let's just take a step back then, because I know this is a mixed audience, and just consider really then what stem cells are. Well, at a very fundamental, basic level, you can define a stem cell as an unspecialized cell. And if you look at them under the microscope, as we have an image here, they're really rather unremarkable. This is a mouse embryonic stem cell. They have large nuclei which contain their genetic information. They don't have any particularly interesting physical features that you look at. And the same can be said for actually stem cells in many different locations. So I think it's important that we understand where some of those stem cells arise from. So where do they arise from? Early embryos is the source of embryonic stem cells. In a moment, I'll tell you a little bit more about that. But we also have stem cells in our umbilical cord, and a lot of people will actually choose to bank those at birth as a potential source of cells, should their offspring require a transplantation at some point later in life for whatever reason. Adult tissues and organs have stem cells in them. So our blood has a tremendous capacity to regenerate. Our red blood cells only last for about just 120 days, so they're continually renewing themselves and turning over. And it's the stem cells that replenish them. The same is true with our muscles. And although people originally believed that actually we didn't have stem cells in our brain because it's very difficult to recover from brain injuries, there is some capacity for regeneration even within subsets of neurons. So stem cells are quite broadly distributed in adult tissues and organs. And we now also understand that stem cells often are at the root or part of the root cause of some particular types of cancer. So there's all sorts of reasons as to why these cells are really interesting, and understanding their behaviors is quite important. I'm going to focus the majority of my talk on or this part of the talk, on embryonic stem cells. So I just want to explain a little bit more about those cells and where they come from. So the top of this image is, it's actually a mouse embryo, but it could be an embryo from any other mammalian species. This is an early embryo, we call it a pre-implantation embryo. It's about 3.5 days old. And the arrow here is pointing to this mass of cells, which is about 40 to 80 cells there. That's called the inner cell mass. If this embryo implanted into a host, that mass of cells would be what would develop the embryo proper. So we know that the cells in that mass has the potential to form a complete organism. Now, biologists and researchers, scientists in general, are curious by nature, otherwise they wouldn't be researchers. And so in the early 80s, people who were interested in development, particularly early embryonic development, carried out a series of experiments of actually taking cells out of this cell mass in this early embryo and then culturing them and trying to work out how they could culture them artificially in the laboratory. And playing with all sorts of different chemical signals and cues and growth factors, etc. And they were able to show that they could indeed take cells out of this inner cell mass, culture them in the laboratory, and mark them in different ways. They could put a genetic marker in them, and then they could insert them back into an early embryo, and then implant that embryo into a mouse and show that those cells that had been from the embryo, cultured in the laboratory, back in the embryo, then contributed to development of that embryo. So that was sort of the first demonstration, if you like, of embryonic stem cells. They are cells that are derived from the very early components of the embryo. They have the potential to form all the cells that make up an adult organism. In mouse, that's over 220 different cell types. And we know that you can take them out, you can culture them in artificial systems, and then return them to their original environment, and they will contribute in a normal way to the development of the organism. So hopefully that quite long explanation gives you a picture of why we call these embryonic stem cells. They're derived from those early embryos. And they have this particular property of turning into all these other cell types, which we give this special name, pluripotent. It means many, potent, that they can change into different things. And we know, and you may not be able to read this lower part, that they can turn into muscle, they can turn into heart, into neurons, into all 220 cell types. So there's enormous potential there. So it's very exciting. And you can understand with that potential that people would get excited by the fact, well, if we can take cells and turn them into particular cell types, then that offers us ways, potentially in the future, of treating chronic diseases that arise due to cell damage or cell loss. And there's just a few examples given here, and there are already efforts underway to, in fact, to do this. So there's big efforts in the US to develop the beta cells of the pancreas that are destroyed in type 1 diabetes. These have potential to be curative treatments rather than managing the disease. They can actually cure the disease. We hear increasingly about the rapidly increasing rates of dementia amongst the older population. So the ability to generate cells that can replace those cells that are potentially destroyed, damaged, either in Alzheimer's or in Parkinson's disease, holds great opportunity. So there's so much opportunity here. You can understand, hopefully, why people are enthusiastic about learning more. But it's not that simple, and research never is. There's a number of different elements that really have to be identified and researched and investigated in order that we can really then turn that potential into reality. And I've just sort of listed some of them here. You have to think about the source of cells, how you can you're going to need a lot of cells if you're going to start treating people. We can do these things on a small scale in a laboratory where we can grow several million cells relatively easily. But when you're treating people, you might need several hundreds of millions of cells for one patient, then if you expand that out to many patients, it starts to give you an idea of the scale. So we have to be able to expand them. We have to be able to turn them into specific cell types and know those cells will actually function as the cell type that we intend. How do we deliver them? Will they be safe? And we have to think about ethical considerations, and I'll come back to this in a little while. So in terms of some of the research questions that I was interested in, I was very interested in trying to understand better how we could manipulate the chemical signals, replicate the chemical signals, which enabled stem cells, embryonic stem cells, to maintain this pluripotent state. So we could expand those cells up in their stem cell condition. And some of the work we did, a lot of the work we did was collaborative across different disciplines. So here, this was a project that I was working with a very talented chemist, Adam Nelson from the University of Leeds, where we were actually using chemical tools to probe the importance of different signals within embryonic stem cells to work out which ones could enhance or indeed interfere with this stemness characteristic, this pluripotency. And this molecule here was a very effective promoter, if you like, of that pluripotent cell type. So, and this is now used routinely to maintain cells in this undifferentiated state. Again, these, you might have, if you remember one of the early slides, we were using this red stain again to show those undifferentiated cells. So these are what cells would look like if we let them grow for 5 days under normal conditions. But if we add this molecule, which targets a particular enzyme, then they stay in this very undifferentiated state. So that's now used a lot to maintain these cells. We were also interested in signals which prevented that maintenance of stemness and actually induced the cells to change and to differentiate. And we found that blocking one particular pathway, and this was an analysis where we analysed lots of different signals within the cells over a period of time, and then did comparison between control and our treated populations, and then looked for differences. And one of the differences we found when we treated cells with chemicals that drove them to change, so they lost their stemness and they became differentiated, we were able to pick out new markers of those that changed during that process. And one of them was a gene that happens to be called ZSCAN4. It doesn't really matter why it's called that. But this turned out to be a really interesting marker. When we first identified it as changing when these cells were undergoing early steps of differentiation, there was very little in the scientific literature, very little understanding of this particular gene. So we discovered something that seemed to be involved in this process that no one else had then identified. And that's one of the great things about research, and that's what you hope to get. But it was a really curious marker because this is a group of cells that they're staining green. That shows you that they're stem cells. And then when we looked at our marker that we'd found, that we know seems to be important in those early cells, it's only expressed by a few of those cells. So it doesn't seem to be in all of them. We didn't understand that. And then we found that if we removed this gene entirely, the cells didn't like it. So that it was clearly necessary, but it wasn't in all of the cells. So it has this really interesting and different pattern of expression that even several years later, researchers are still trying to understand why and why it's important. But we do know this gene is important for maintaining the stemness, and it seems to do that by maintaining the integrity of the genome in those cells. So it seems to be really important, but exactly how it does it, we haven't yet worked out. But it was fascinating, and then it came out of this study. And I suppose people often ask me, do you regret not doing research any longer now that you're working in a different environment? And I suppose when I was looking back at this and I was just checking in with recent publications, I was thinking, I do kind of regret not, no, but you can still follow the research through, but so a little bit. So that just is a little bit of a taster, really, about some of the signals that we started to unravel that were really important in embryonic stem cells, both in those early stage cells to keep them pluripotent, undifferentiated, but also the steps that then they took when they were changing into other cell types. And you won't be able to read this, but the key point is that there are many, many different signals that we now understand, and our work contributed to this, that actually are important in regulating the behavior of those cells. And as with everything, the more you research, the more complex it becomes and the more layers there are. So there's still a lot more that we actually need to understand. But I just wanted to touch on two other sort of projects that I was involved in. So this comes back to the point around expansion of cells. So it's very important, actually, that if we are going to realise the potential of stem cells, that we have ways of actually expanding them in their stem cell state, or because when they become differentiated, they tend to stop dividing. So there's limited capacity for expansion of very differentiated cells. So you have to kind of expand at the earlier stages. So this was a collaborative project supported by the organisation that I now lead in the Engineering and Physical Sciences Research Council. So this was actually a collaboration with chemical engineers, where we were looking to actually try and develop sort of three-dimensional systems. This is this is a spinner flask, which is a three-dimensional system. Normally, we would just be growing the cells in 2 dimensions. But 3 dimensions offers obviously greater capacity for expansion in similar volumes. And this is just a video showing, so we'd used these, they're gelatin-based hollow spheres, the cells that the embryonic stem cells liked to grow on and sorry, I put that down. Oh, it stopped going. I'll have to go back and start it again. No. Oh, no, it doesn't want to go back. Okay, let's try that again. Oh, I'm sorry. The video just done it. It was supposed to be on a loop. No. Well, anyway, it doesn't matter. So what we do, we were growing the embryonic stem cells, and it worked for both mouse and human, on these kind of gelatin spheres. And the gelatin spheres in the grey, and the blue shows the cells that are growing around the outside of the sphere. So you're increasing the surface area hugely. And we were able to show that the cells, we could develop conditions that actually the cells would maintain their pluripotency whilst growing on these particular spheres. They maintained their metabolism. We did have some that they really didn't like, and they would just die. So we actually then sort of started to move forward to define conditions which could actually be used for expanding these cells up if you wanted to increase their numbers. So that was a little bit about sort of a snippet around expansion. And then some other work that I was interested in, and is still ongoing in the laboratory in Bath, and I check in with it every now and again, although I don't have any formal responsibilities there anymore, was to start, we became involved in a project which was supported by an organisation, Stem Cells for Safer Medicines, which was about trying to use human embryonic stem cells to develop hepatocytes, which are the major cells in the liver. Now, for those of you who may have been involved in the pharma industry, if you're developing new drugs, one of the biggest problems is toxicity in the liver. And the models for testing drug safety are not very predictive. So, and human hepatocytes, if you derive them from donor livers, don't maintain their characteristics for very long once they're in artificial conditions, and plus they're in short supply, and the livers will be used for transplants as well, so there isn't an infinite source. So this project was about how we could use the potential of stem cells to address this challenge of developing liver cells that could then be used for testing medicine safety, but also if you get functional liver cells from stem cells, you could imagine then how you could then use those within a transplantation type setting as well for treatment of patients. So this was a project looking at that, and if you just look at the kind of the images down here, we've taken the cells and we've tagged them with markers that we would expect to see in liver cells. So the green is showing the markers to indicate that they have changed from the stem cell through this protocol. Quite a long time. It takes about a month to turn the cells from a stem cell into a liver cell. But we start to see that phenotype coming up, and they start to function like stem cells. As I said, this work is continuing, and not just in the lots of people are interested in this particular type of problem. So that's so I now sort of want to move on a little way. You will all recognise this lovely sheep here as Dolly. And it's kind of an interesting segue. So Dolly was created in 1996 by scientists at the Roslin Institute, who were supported then and continue to be supported now by the organisation that I lead. So there's a dual purpose for this. So we all know who Dolly is. Now Dolly was amazing because she was the first animal to be produced from a technique called cloning. And she was the inspiration for this Japanese scientist, Shinya Yamanaka, who was interested in the fact that you could reprogram an adult cell into an embryonic state that you could then implant and generate a healthy mammalian organism, i.e. Dolly. So he was really interested in that. How amazing was that? How did that happen, that whole reprogramming from an adult cell right back to something which had the potential of an embryo? And this was at a meeting that was actually at the Roslin Institute a couple of years ago. It was the 20th anniversary of Dolly's birth, the 5th of July, 1996. I know that because my daughter was born a year later, so we always remember the date, the 5th of July. And he was very clear that Dolly was an inspiration for his work, for which he was awarded the Nobel Prize. So what was that piece of work? So he said, okay, if I take adult cells and I put different factors in them that I know from my research and the research of others that are important for that pluripotency of embryonic stem cells, can I turn those adult cells into cells that look like embryonic stem cells? Now, if a grant had come in on that to our organisation, because we support research across the board, without any evidence to support it, we'd probably have said, that's a bit of a crazy idea, I'm not sure that's going to work. And lots of people did. It was so simple, it was such a simple thing, but he demonstrated it worked. And I remember being at the conference, it was in Toronto, where he did the big reveal, and you could hear there were gasps kind of around the audience, because people really thought, fantastic, this was conceptually so simple, but also so challenging at the same time. So what did he do? He took adult skin cells, actually, the first thing that he did, and he only needed to introduce 4 factors into those cells, and he switched them from being adult skin cells into cells that looked and behaved like embryonic stem cells, and he called them induced pluripotent stem cells. He first described this in 2006, and then in mouse, and then in 2007 reported the same findings in human, and then he was awarded the Nobel Prize in 2012 alongside John Gurdon, who was a British scientist working in Cambridge who'd actually done the original experiments on reprogramming in frogs. So that's a really short period of time, 5 years, between a discovery and a Nobel Prize, so that tells you something about the significance of this. Why did he do it? What was intellectually interesting, but actually also in Japan, there were the ethical concerns around using embryo-derived cells for any potential treatments. And that's the case in many other countries as well. In the UK, we have quite a broad regulatory framework which allows research to continue. But in some nations, and there were particular issues in the US as well, people were not permitted to use human embryonic stem cells derived from human embryos because of the ethical concerns. And that's something which I'm quite happy to debate with you, but that was the situation. So he was looking for ways of which you could have the potential of embryonic stem cells, but without the ethical considerations. But there's more than that, because actually, if you think about it, if you're a patient and you're suffering even from a rare disease or a common disease, this technology means that researchers can take cells, a skin cell, it's easy. The technique's been used with many other cell types as well. Introduce these factors and generate patient-specific stem cells that can then be used for a whole host of different studies. They could be used for generating cells for replacement into that patient. They would be matched. You wouldn't have the difficulties of rejection that we see in other types of transplantation. If it's a rare disease, or if it's a common disease, these induced pluripotent stem cells are now showing how they're being used for research, but also by the pharmaceutical business to understand disease mechanisms, because you've got human cells that you can study that have the genetic basis of these diseases. And people are doing that, and there's been some really interesting examples of using induced pluripotent stem cells to study cardiac disease and to start to identify new potential treatments to control different diseases. Disease in a dish is a particular term that people like to use. And it's opened up a whole new world of opportunity, I would say. And this is why it's so significant, and I think why the Nobel Prize was awarded, because of this potential, because we now have routes to doing things that we could only have dreamt about in previous times. So I'm now going to switch gears a little. I will come back to stem cells. I will have a little bit more on stem cells. But that was sort of, that was kind of the background. And it was a really exciting time during that sort of period when I had my fellowship and then beyond that. And it really transformed things and really sort of changed things. My fellowship meant that I had the freedom. I wasn't tied to the academic year, which has a particular pattern to it. And I was free to travel, to get involved in different things. I was involved in a big European program, and that involved a lot of travel, going to meetings. Was lucky enough to go over to Japan. So I'd hosted Shinya Yamanaka at a meeting in the UK before he was famous. I say this, and then he returned he invited me over to a meeting in Japan, and actually my husband came along with me. So he dines out on this story that I've met this Nobel Prize winner and had various types of sushi, and he thinks that's great. He's an environmental engineer, so he doesn't have that same excitement around research that I do. But anyway, or maybe he does. I may be doing him a disservice. I think the great thing is actually having been involved, knowing the people, and he's a genuinely fantastic individual who really wants to help humanity, and I think his research is really doing that. So research was going fantastically well. So you could probably be thinking, why would you then sort of want to change direction? And I think I've only worked out later in life that actually every few years I like to have a new challenge and a new change. I get a bit too comfortable, perhaps it's when I get a bit bored with doing things, so I like to have something new. And so that change of direction came when I moved to Bbsrc. And I was approached I was headhunted essentially, would I be interested in becoming Director of Science? I've added a little bit to this kind of diagram here, my promotions, and I'd become quite involved in Bbsrc, both through my fellowship, and I was involved in some of the panels and committees and things that we had. And I thought, well, actually, I've always been really interested in kind of broader areas of science, and here's an opportunity to actually get involved in an organisation that supports British bioscience across the board, and it's an opportunity to sort of work at a national level and have a wider impact, I suppose, not just in my own very narrow and deep area of research, fantastic though that was, but that sort of broader piece. And there's a sense also of sort of supporting the system that had supported me and helping to really develop and deliver on that potential. So I went as Director of Science. I then became Interim Chief Executive when our former Chief Executive left a little bit before, she resigned before the end of her term. We were just coming up to the creation of UK Research and Innovation, so I was asked to take on the role. I'd never imagined that I'd be a Chief Executive of anything, here I am, and then I became the first executive chair of Bbsrc when we joined UK Research and Innovation, as Rod introduced. So it's about sort of new challenges, and I always show this slide when I talk to our constituents, to our community, and universities and institutes, because I think it's really important that we remember that UK Research and Innovation and Bbsrc within it are funded by ourselves, the British taxpayer. And we receive our funding through the Department for Business, Energy and Industrial Strategy, and obviously Treasury. And the money that goes into Treasury is from all of us. So there's an imperative for us to make sure that we are sharing the knowledge, the outcomes of that research for the benefit of the taxpayer. So what Bbsrc does, and you can understand why I refer to it as Bbsrc, because Biotechnology and Biological Sciences Research Council is just far too long a mouthful. What we do, we want to support world-class research in the UK, in our universities and institutes. And we have a budget of about 450 million a year. So we have significant investments on behalf of the UK taxpayer. We support the training of the next generation of scientists. That's so, so important. And we want to make sure that the research gets out of the lab and has impact in people's lives and drives the economy. It's knowledge made useful is one of the terms that we refer to. And because we are supported by taxpayers' funding, it's important that we have public support and public dialogue such that we can exchange and we understand. Bioscience, we do work in areas that are potentially contentious. Stem cells is one of those, and I'll come back to that at the end. So we have sort of these four aspects, really, four aims for our organisation. And we're interested in maintaining bioscience as world leading, because by many different metrics, and this just happens to be one, doesn't really matter what it is, but all of those that are ringed in green is where the UK is the leading nation, and many of those are bioscience related. So we do actually lead the world in research in the area that we support, and we want to keep it that way. And the way we approach that is that we have a strategy. We are always horizon scanning. What are the next big areas that we need to be investing in? What should we be supporting? Particularly across the disciplines. And this is where coming together across all of the research councils in UKRI offers so much more, because it reduces the barriers, really, for that multidisciplinary collaboration. And hopefully, as you can tell through my talk, I've benefited from those interdisciplinary collaborations. And there's so much more that we can do, things cut right across. So we're interested in advancing the frontiers of knowledge. Bioscience has some of the answers, and I'll illustrate some examples in a moment. We're tackling some of these big societal challenges. How are we going to feed the growing population whilst at the same time not reversing loss of biodiversity, tackling environmental and climate change? We know that agriculture is one of the biggest polluters. How can we turn that around? We have to. We absolutely have to. Bioscience has a really important role to play there. Bioscience also has a really important role in developing new bio-related processes and products to replace those that currently we derive from fossil fuels. Biology is amazing. Look around you. It does amazing things. We can harness that to actually turn into new processes and materials and chemicals and fuel. And clearly, understanding health and health of the wider environment, of animals, and how disease can transfer, zoonotic diseases can transfer from animal to man, antimicrobial resistance, some really big challenges that we're facing as a society. Bioscience has some of the solutions to help address those. And of course, we can't do any of that unless we have the people, we have the infrastructure, so biologists by and large don't tend to use the Large Hadron Collider, but we do have other large facilities. So in the UK, we have the Diamond synchrotron, which gets used an awful lot by biologists because we're interested in looking at sort of molecules, structures, and so we might not use the LHC, but we do use other sort of large facilities. So really important we have that. And working in partnership and collaboration and making sure that we're effective at exchanging knowledge is really important. So we have a lot of international collaborations as well. And research, it doesn't respect boundaries and borders. People will want to work with the best researchers wherever they might be, and we have ways of supporting that. And I just want to take then through a few examples to illustrate some of the types of discoveries that are broader than stem cells, although the first two are on stem cells, that we have supported. And so it's really important, and this is just to give you a flavour of the breadth of the research and the opportunity that there is. So this is research carried out by Alfonso Martinez at the University of Cambridge, where they have actually used embryonic stem cells, and they have demonstrated that if they differentiate those embryonic stem cells towards different cell types and then bring them together, they can actually, the cells self-assemble and essentially form artificial embryos. So this is actually really, really quite surprising in some respects, but really interesting because it potentially means that we can reduce the number of animals that might need to be used in research, because you can actually generate these structures in the laboratory and can study them in that way. And it actually also has interesting applications, potentially sort of in prenatal medicine and in fertility research. So again, quite unexpected finding, quite fundamental science in stem cells, so I always look out for those particular ones. And this is actually a plant stem cell. So this was a PhD student at the University of Oxford who was looking at samples in the herbarium. So they have an amazing collection at the University of Oxford in their Department of Plant Sciences. And he was looking, I think it's a 320 million year old sample, a fossil. And he'd been studying ancient plant roots and looking at sort of trying to predict that development. And the structures that he saw in this ancient fossil were almost identical to structures that he would see in plants that were living today. So it just kind of gives you that whole sense of evolution over time, and clearly has been a mechanism that has worked. So these were the oldest plant root stem cells that had been identified. And it was just a sort of a serendipitous discovery, but actually quite significant, thinking that's a mechanism that has been maintained sort of throughout that evolutionary period into plants that we know today. I don't know whether any of you like whiskey, but this is actually a study that started as a very fundamental study about trying to understand how plant cell walls were constructed and the different processes that went towards constructing those cell walls. And the researchers at Rothamsted Research had used a model plant to look at this and to understand this. But it was one of the issues with the whiskey industry is that they don't like UK-grown wheat because it's too thick, it's too viscous. And it's viscous because when you actually make the extracts, some of the plant cell wall material, the arabinoxylans, if anyone's chemically inclined, kind of are contained within that extract, and they give it viscosity. And this causes a problem for the distillers because it clogs up the pipes and it costs them more to clean, and so they kind of don't like using them. So they'll import maize and other sources for their distilling. So that's not great. That's a missed opportunity in the UK, and why wouldn't we want it to be the UK? So what these researchers have done is that they've translated their findings from their model system, and they've identified those same sort of pathways. They've generated, by conventional breeding, new varieties of wheat where the viscosity of the extract is reduced by about 70%. And so the distillers think this is fab because it actually is very similar to what they would use anyway. They can use UK grain, so they don't have to import grain, and problem solved, you could say. But this is an example of some very fundamental research about understanding fundamental processes of plant biology that actually then find application in perhaps unexpected ways in different areas. Climate change is one of the biggest issues facing us today, and we hear a lot about the greenhouse gas methane emissions from cattle, and there's quite a lot of debate around that in society at the moment. And researchers that we support, again at the Roslin Institute, the University of Edinburgh and Scotland's Rural College, have been looking at the links between kind of the genetics of the cow, the microorganisms that are in their guts, which are responsible for the methane production, and whether actually you can select cattle that have that their genetics means that the microbes in their gut produce less methane, so that you actually then are generating cattle that are naturally lower producers of methane. And in fact, this study showed that there is a link between cattle genetics, their gut microbes, and methane production. So if you could actually select for those low methane emitters, you would actually really start to address the problem of greenhouse gas production from our cattle industry, not just dairy potentially, but also from the beef sector. So quite potential for broad impacts there. Lots about plastics, single-use plastics, trying to avoid single-use plastics. I'm very pleased that there's glass bottles here today. So one of my New Year's resolutions was to reduce the amount of single-use plastics. Last year we had, the finding was published, led by a group at the University of Portsmouth in collaboration with colleagues in the US, where they had identified a bacteria in a landfill site in Japan. I'm not quite sure why they were sampling the bacteria, and I haven't got that part of the story. But these bacteria had adapted so that they could use the plastic bottles on the landfill site as their source of carbon as energy source. So this is quite curious. So the researchers, being curious individuals, said, well, what are the enzymes that must be breaking down that plastic that are then allowing them to release the carbon energy source, allowing them to multiply and live? They identified that enzyme, they improved the enzyme, it can degrade polyethylene tetraphthalate, which is the major constituent of those single-use plastic bottles that we all have. I can see one in front of me there. That might be. This enzyme, they've improved it, they've adapted it. Potentially, it can help in degrading plastic waste, recycling the component parts to actually then sort of get away from sort of a single use, much more to a circular use of that plastic. So this caused great excitement when it was actually published, and it just shows the fantastic things that bioscience can do. And another one is around coffee waste. So Colombia is the third largest producer of coffee, and researchers at the University of Surrey, through an international program, had been working to try and develop systems whereby they could alleviate the problem of pollution from coffee waste, because the coffee producers are typically smallholders, typically in rural areas in Colombia. Though they have no way of kind of cleaning up the water, and coffee requires quite a lot of water in the processing. So they were just dumping that into the rivers and the water courses, causing pollution problems. So how could they actually solve this problem? You kind of have to think about ways you can use the waste. What they've actually worked out, a way of doing, is building a microbial fuel cell, and the microbes in that fuel cell essentially use the coffee waste as their source of energy, and they produce electricity as a consequence. This is an image of it. They're fairly cheap to actually put together, and they're now sort of looking to roll this out onto smallholder farms in Colombia. There's benefits, less pollution, an alternative source of energy, and sort of using a waste stream, so there is no longer a waste stream. So this was actually awarded a prize at the end of last year for this particular research and the potential for this particular type of technology. And that was a collaboration between bioscientists and engineers. Bioscientists don't tend to build these types of things. You need a really good engineer to help you do that. And we do a range of, support a range of things, and we like to recognise the researchers who are sort of leading in the world. So when we had our 20th anniversary in 2014, we had our anniversary awards, and these are three of the researchers here. Russell Foster, who works at the University of Oxford and he's very interested in sleep. studying sleep, not sleeping himself. Caroline Dean, who works on plants and how plants sense temperature and why that's important. And Jeff Errington, who works on, who's an eminent microbiologist and bacteriologist. And I included this slide because Pallab Ghosh was actually at the awards. And when I was an undergraduate at Imperial, and I don't know if anyone else was, he was the editor of Felix, which was the student magazine. Is it still the student magazine? I don't know if it still is. So it's just kind of interesting that there's that kind of connection there, and obviously he's well known to us now. And we also I talked about realizing social and economic impact from the research that we support. And we actively encourage our researchers to think about how they can take their discoveries out of the laboratory and really translate and have impact. And this is our Innovator of the Year, actually our 11th competition. We are hosting the awards event tomorrow evening at the Science Museum. So right about now, we will be announcing the winners, and I will be hosting that event not very far away from here tomorrow. And we've had some great winners. The winner last year was Rob Honey from Cardiff University and Sabrina Hatton-Cohen. They had taken studies, so Sabrina is a firefighter, and she had been interested in behavioural neuroscience and decision making and how she could use what she discovered through her PhD that she was doing part-time, which is amazing, how they could actually use that and embed that into decision making in firefighters, because they knew that this was one of the key problems that the incident controllers had. How do you actually make decisions, and sort of having checklists, and by taking that sort of behavioural neuroscience view, they were able to improve this decision-making. It's been adopted as guidance in the UK, and people are interested in it internationally. So you can have impacts in all sorts of different ways that you just can't imagine when you start your research. But that's part of the fascination of the role that I have now. I mentioned public dialogue, really, really important, particularly in the bioscience when we're dealing with areas that are potentially quite contentious. It's important that we actually have a debate. I was involved in this piece of work that Bbsrc did with the Medical Research Council when I was a full-time researcher in Bath, the stem cell dialogue, which involved being immersed for 2 days with a group of individuals for a cross-representation of the demographic in Bristol, as it was at the time, and sort of debating this and looking at sort of the issues and looking for that public support and things that we as researchers needed to be paying attention to, actually. And there's many other examples around this as well that we've done. So we take this quite seriously, and I think as UK research innovation, we have an opportunity to do that much broadly across the piece. Public engagement is really important. As part of our 20th anniversary, we hosted a great British Bioscience Festival at the Museum of Childhood in Bethnal Green. We had amazing exhibits. It was so busy all the 3 days that we were there. We brought in a huge range of people from a huge range of different backgrounds and helped them to understand and hopefully share a bit of the excitement about what bioscience can actually do and offer. And as part of the training that we have for students, our PhD students that we support, we give them the opportunity to spend 3 months doing something outside of the laboratory, so having a different type of experience. And this particular student, who was based at the Royal Veterinary College, actually worked as part of the Royal Institution Christmas Lectures. So UKRI and Bbsrc have supported the Christmas Lectures over the years. I've been to the filming of them once, and it's actually quite a fascinating thing. But to actually work on the production of that and to bring science to life, as it were. And this is something, obviously not every student does this, but we encourage our students to look up from the laboratory bench or from the computer, whatever their research might be, and actually to get these different experiences to do different things that they might be interested in doing in the future. So I'm going to stop there. It was almost spot on 8 o'clock. And I think I'd just like to finish up by just sort of summing up. I know that's been sort of a talk of 2 halves, but I think hopefully the theme that runs through it is how amazing science is broadly, and obviously I'm biased, but bioscience in particular, the potential that we have for stem cells and for the future around that, about how I think that it's important to recognise opportunities and to take those opportunities when they come up. There were things that when I was here at Imperial I would never imagined doing, and there's all sorts of reasons that you can convince yourself not to do them, but sometimes by doing them and taking that step, actually that's sometimes the best thing that you can do. So just encourage people to seize opportunities and see where it leads you in whatever field that you might actually be. And I'll finish there. Thank you very much.
Thank you very much.
Thank you.